All right. Hello, everyone. Welcome to another session of our evolution seminar series. So today we'll be thinking and learning about phylogenetics, the phylogenetic networks in particular. So we're joined by Professor Cécile Ané from the Departments of Statistics and Botany. So without further ado, let's get to the science. Please join me in welcoming Professor Cécile Ané.
Thank you very much. I'm really happy to be here. This seminar series have been one of my favorites for as long as I've been here. And this is where I learn about the breadth of evolutionary biology on campus and beyond. It's really great. So thanks for the invitation. Okay. I'm going to talk about phylogenetics. And I'm going to start with a paradigm shift to explain why we are interested in networks. And then I'll be talking about the pros and cons of networks. And if I should have some time to cover how we use then these networks to look at trait evolution. So this is a paper that came out in Nature 2003. It was a big paper at the time because at the time people typically had one gene or two genes to estimate their phylogeny. And this paper had 100 genes, 106. And it was like a big thing. And Anthony Srokas, he was here. He presented these results here at this seminar series. And so they had many genes, 106. And their findings was there is a lot of discordance between these genes. They don't all tell the same story. They give us different trees. Something that they say incongruence between these genes. And then they said, oh, well, if you just concatenate all these genes, you get this one tree with really good confidence. Bootstrap values are all 100, posterior properties are all 100. You really get this high confidence tree, and so it's all good. And then they did some simulations and said, you really only need 20 genes. So if you only have 20 genes, you're good. So it's like, oh, that's very cool. We get more data, we get more confidence. This is great. So this is ignoring the issue of incongruence, really. But what can we do? So the next 20 years later, you think that we have way more data and that we have really good solutions by now. Yeah. So yes, we do have way more data. We have next-gen sequencing, lots of, targeted sequencing, RAT-seq, lots of different kinds of data to get phylogenies. And then I picked this paper because Anthony Srokas is also on this paper, it also high profile. And basically they say, well, incongruence remain. We have thousands of genes now, and we still see a lot of incongruence. Depending on the gene we look at, we get a different tree. And they say it's hampering our ability to get the tree of life. Well, I disagree. That's my emphasis. I think instead of hampering our ability to know something about the past, it gives us more information to know something about the past. So it's the same paper, and they say, well, here are reasons why there is incongruence. Now we understand better, so 20 years later, we understand more. So one reason is incomplete lineage sorting, and I will show what this means. Horizontal gene transfer, that's something you might be more familiar with, hybridization, introgression, recombination so that different genes have different stories, and other reasons can lead us, these genes, to not have the same tree. And then they say they differ from the species tree. And this is where I think, even though this is a 2023 paper, it's not quite capturing the paradigm shift. Because the paradigm shift is, well, look, we should not look for the species tree. A correct depiction of this ancestral history is not a tree. It's really a network where we can describe all these events. So incomplete lineage sorting, this is happening everywhere. That's a process that's within a population, including ancestral populations. These processes would be migration between populations without changing the number of species. Hybridization would change the number of species in that it would be one more. There could be species mergers. Let's say you take a boundary that separated a species that then diverge, and then the boundary goes away because glaciers melt, and the two species come back together and merge again. That will also change the number of species from 2 to 1. But all these can be represented on a network. And there are other reasons not cited by this paper that would be gene duplication and loss if we don't know that this is happening. If we mistakenly think that all the gene copies we have are orthologous. And I'm not going to talk about this. We don't have all the answers yet. All right. So to show you what a network might look like, this is one from Nisa Karimi and David Bell back there on bailouts. Most of them are in Madagascar and one species in Africa and some species in Australia. All right. So here it's mostly a tree, but this is this extra edge that is still a hypothesis. We're not completely sure, but that would make the tree not a tree and more a network. Okay, and that would represent gene flow from this species. It would be ancestral. It's passed into that species. So this lineage, I don't know past back then, but today these are in Africa. And this lineage, I don't know when, but all these are in Madagascar. So this gene flow is surprising because it's jumping geographically. So that's one example. There are in the literature many examples of much more complicated networks. And many of these are in high-profile journals, Nature Science, and especially if they are from human populations. So here, we have a few modern human populations, and all of these tips are ancient humans. This is 2.8 thousand years ago, 3.5 thousand years ago. So these are pretty ancient. It's really wonderful how we can get DNA from these ancient bones. And then that's a complex network. All these arrows, like this network says that this Nepal population that is 2.8 thousand years ago had genetic material from 2 ancestral populations. So it's a mixture, or it's a hybrid, if you want. So we don't really know what happened if it's, well, yeah, but it just says 16% of the genome came from there and 84% of the genome came from there. So I would argue that this paradigm shift is don't try to get the species tree, but get a more detailed picture of what happened that would be captured in a network. So let me talk about networks. What are the methods? What information, where does the information come from? And then talk about the pitfalls. So what is difficult? So this is the type of network you might see in many publications still, and because it's easy. The methods to get these networks are fast, and these are still useful. These networks are implicit, meaning that these networks, they describe the incongruence between genes. They describe the mix of, of information that we get. So here, for example, for the most part, all these virus strains group together as a clade, but there is a little bit of incongruence. Whereas how these clades relate to one another, there's a lot of incongruence, but it's not telling us about the past. It doesn't explain why we have this incongruence. It just describes. And these networks have been used for a long time. And it's more recently that we've been using explicit networks where each node represents an actual ancestral population. Each edge represents an actual lineage evolving. And then a node like this would be called a hybrid node because it receives genetic ancestry from two parent populations. And these networks don't say what was the process. They just say this population has a proportion gamma of its gene coming from here. And so then the proportion 1 minus gamma coming from the other side. It doesn't say whether at this point in time, there used to be 1, 2, 3 populations, and there was gene flow. Or perhaps there was just 2 populations, 1 and 2, and these 2 hybridized and created a new one. It doesn't say, that network does not say anything about the biological process. It just says, this is the result in the end. You had this population with mixed ancestry. So this gamma is really useful if we think of gene flow. Perhaps we're thinking about here the Neanderthal population and here modern humans. Then this gene flow would probably give us a proportion that's pretty low, like 2%. If this is a result of hybridization where we actually create a new species, then this gamma might be closer to 50%. Okay. And so this network would be explaining part of the incongruence between genes. So if this is the only thing that happened, then if I trace the ancestry of an individual from population C back in time, here there is some probability gamma that for one particular gene I picked, the ancestry comes from here. And so this gene would look like this. We'd have a tree that looks like this. And another gene might have another, because of recombination, could have another tree. And with probability 1 minus gamma, it would have this tree. So that explains part of the incongruence. Unfortunately, there are many other reasons for incongruence. And the one reason that researchers have focused more initially was incomplete lineage sorting. So what is this thing? And this would explain extra incongruence. And it explains why it's actually pretty hard to get these networks. And so I'm going to try to explain this incomplete lineage sorting. So imagine this is one population. Time goes forward from top to bottom. And with the Wright-Fisher model, we assume that at each generation, so each row here is one generation, and each little dot is one individual. And so I'm imagining a population with a fixed population size. At every generation, I have the same population size. And then if it's all neutral, at the next generation, each individual picks its parent from the previous generation at random. So this individual would pick this parent, this individual could pick the same parent, and so forth. And so that goes on every generation. Each child picks a parent at random from the previous generation. And at the end, we have the population that we can observe today. We sample a few individuals. It could be only one. It could be a few, 4 individuals. Like here I have 1, 2, 3, 4, 5 in blue. And then I can ask the question, so these individuals, for this particular tree that I'm looking at, for this particular gene that I'm looking at, what is its ancestry? What is its genealogy? And so at each generation, I can ask the question, did these 2 individuals came from the same parents? Well, no. So previous generation, did they have the same parents? Well, no. And go back in time like this. So we don't have any data on the other individuals, but we can get the gene tree for the individuals for which we have DNA. Right. And that might look like this. So if we use this model of just random picking of the parent, we can predict how long it takes to find the first common ancestor between my sample of individuals. And it's described by a nice exponential distribution. So if you don't like probability, you don't need to know and care about this, but it's really easy to describe mathematically. Okay, all good. This is what happens in one population. It's just about genealogy. The thing is, we typically think we don't need to worry about this process when we're thinking about long evolutionary times. And if we only pick one individual per population. Well, we do, because when we trace the ancestry of this particular individual, I'm looking at a particular gene that's from chromosome 1, whatever, and I look at its genealogy back in time. Here, when I'm reaching this ancestral population, I'm looking at 2 individuals now. The 2 individuals that are the ancestors of the ones I sampled. And so they don't have a common ancestor right away because they're in the same population, and we need to look at the genealogy of these 2 individuals within that population. And if the speciation times here are very one right after the other and don't have much time, it could be that they don't coalesce, they don't have a common ancestor right away for this gene. So that's just, it could happen for some probability, some proportion of genes. And then when I reach this even more ancestral population, then I'm with 3 individuals. So here it is possible that these 2 individuals have a common ancestor from their genealogy. You know, in orange here. Oh, I guess people in Zoom. And then, but then that's it. No more common ancestor. And then we hit the most ancestral population here. And it could be that these 2 individuals have a common ancestor first. And so this gene tree, then the only thing we see when we sequence the DNA is the tree for that gene. The tree that has colors here. We don't get to see the big fat tree, right? And we would see, oh, C and D, the group, versus A and B, right? So it's the same topology. On the right-hand side here with probability gamma. So this tree that could happen because of reticulation could also happen just, I don't know what I should be doing here. Okay. Yep, went away. Okay, so C and D are sister in some genes, maybe in a small proportion of genes, but in some, and not because of hybridization. Only because of this process of what happens within a population called incomplete lineage sorting. And so in methods that try to estimate these networks, we need to account for this process of incomplete lineage sorting. Otherwise, we might want to be adding edges in a network just to explain this process. And we don't want to be doing this. Okay, so there is now a whole lot of methods that will take DNA and estimate a network. And so some of them ignore incomplete lineage sorting for various reasons. Maybe for viruses, that's not as relevant because they're, yeah, I don't know. For computational reasons, for sure, it makes things easier. And then there are many methods that do model the incomplete lineage sorting on top of having reticulations in the network. So some of them use SNP, basically, and allele frequencies like tree mix. Maybe many of you have heard about tree mix. Admix tools 2, This pool F-stat, I haven't seen it used very often, but it can use pool seek data. So this is really cool. Admixture based, so really accurate, but slow. Mommy2 is really great. And then these are more using these are more at the population level within species, and these are more for between species, so deeper in time. Some of them will use gene trees as input rather than SNP data. So various methods in PhyloNet. SNAC is a method I worked on, and it's integrated in a package that can do a lot of things for networks. And various Bayesian methods in that are accurate too. So we're at the point where it's unclear which method is really best. What's clear is that some methods are very slow, and that's why they're not used. Oh, I forgot to mention BPP also. Oh, I know. That's because it doesn't estimate the network. It will use a fixed network, but not estimate the network. Really, the methods in there that are used the most are the ones that run faster, basically. And all of them are limited in the number of taxa that they can handle. And some of them, you give them 5 species, they can run, and then 6, it's really slow. And so that's limiting. And so there is one method in PhyloNet that used fairly often, and SNAC is used often because they scale best. And then tree mix also scales really fast and is being used very often. Okay, so the next thing I wanted to explain is where is the signal in the data about these hybrid events? So the signal here comes from an imbalance. You know, how do we tell apart? Basically, how do we tell apart that a gene tree that has C and D sister is a signal for admixture versus no, it's just incomplete lineage sorting? How can we tell? And basically, it's the imbalance. So in this scenario at the top, what I'm showing is these events that happen fairly rarely. I mean, it depends on the speciation times here, but incomplete lineage sorting where they don't coalesce, and then later it looks like the wrong species are grouped together, right? And when that happens, it happens in a way that is symmetric, in that if there at the SNP level, at the DNA level, if we see that this species T3 shares the same SNP as species T2, that's like, oh, is that a signal for admixture or reticulation? Well, it would happen with the same frequency that this species T3 shares the same SNP as species T1. If this is only caused by incomplete lineage sorting, the frequency of these two things should be the same. And so we would be tempted to say, oh, no, this is just incomplete lineage sorting. We can explain this just with incomplete lineage sorting. We don't need to add gene flow. If the frequencies are different, if T3 groups with T2 more often than it groups with T1, so T3 and T1, we have the same SNP, rarely, but this more frequently, then that would be, oh, there is an imbalance. So I want to group T3 and T2 using some hybridization events. So that's the basic source of information for that phylogeny to not be a tree and be a network with reticulation. I'm happy to spend more time on this. So we can look at this in terms, so this is really looking in terms of DNA, where we have species that share the same site, the same SNP versus others. But we can look at this in terms of gene trees. For each gene, we don't look at only one SNP, we look at the whole sequence of DNA, and we get a tree for that gene. And then we can look at the frequencies of these gene trees. So now we can also not just look at 4 populations only. Here I'm only looking at 4 populations in our group and 1, 2, 3 populations, but I can have more than 4. And in that case, what we would want to do is look at all subsets of 4, okay, and then put them all together in one big network. So that might be here on the left, a candidate network. And I want to know if that network fits my data. Okay. And so what I can do is look at all subsets of 4 species I get from it. So for example, A, B, C, D. If I only focus on these 4, from the candidate network, the relationship between these 4 species is what we have on the right, where I don't care about E, so I can erase this, I can erase that. I don't care about F and G, so I can erase this and this whole thing. And so from here, I can ask, what do I expect the gene trees to look like? And what frequency? So if here, at this ancestral population, 90% of the genes came from here, and 10% of the genes came from there, well, most of my genes should have AB versus CD. And this is what I denote by AB/CD here. And if there's no such, if my candidate network is not good, if in fact I should have a tree, then I would expect that the other 2 options, so in A, instead of grouping with B, it could group with D or it could group with C. There are only 2 other options. Okay. A, if it doesn't group with B, then it groups with C or with D. And if my network, my candidate network is not a good fit, if it's wrong, if it should be a tree, then I would expect the other 2 options to be equally frequent. Just like with the SNPs. If it's tree-like, the frequency of these surprising patterns should be equal. It's the same for gene trees. If it's because of incomplete lineage sorting, these 2 surprising gene trees should occur with equal frequency. So that's what I'm writing here. If there's no admixture, if it's just tree-like, these 2 frequencies should be equal. And the signal for this edge comes from the fact that A and D, meaning B and C together, they occur more frequently than the 3rd option. Right? So this option groups B and C together. B and C. That's what I would get from here. So these methods use really the symmetry and the frequencies to get a network that fits as best as possible using all subsets of 4 taxa. So this is also what's done in admix tools, using the F4 statistics. Maybe you've heard about the F2 and F4 statistics more than quartet frequencies. So we can search for a network that has the best fit. We can also use a candidate network and ask, does it fit well? And here, maybe I should pass more quickly because that's more of a statistical question. We would look at all subsets of 4, see if this subset of 4, if the data for the subset fits the little network of 4. And then there are lots of subsets of 4. So if I'm looking at a network that has 20 taxa, that's a lot of subsets of 4. And so it's an issue of multiple testing. Tests are not independent. So that's an interesting statistical question that I worked on. So here's an example just to show that it's starting to get towards the pitfalls. So it's the same study by Nisa Karimi and David Baum. We had a number of networks that seemed to be not bad. So hypotheses. And so I could look at, do these fit? Okay, these are the networks that we obtained searching for the best networks. But then, do they actually fit the data? And the answer was no. No, but they're still the best. So it's like everything in statistics, not all models are wrong. Just some of them are helpful. So they don't capture everything about the biology, but they probably capture something real still. What was interesting was if we excluded these two or these two, which are here and there, then the smaller networks do fit the data. So what we're missing in these networks is it could be something about these taxa. So it's and also now another pitfall is if I remove these two, the Adansonia ZA or Adansonia carrieri, I get a smaller network and they both fit, but they're different. So it's like if you're confused about confidence intervals, there are like two networks that are part of a confidence set of good networks. We still don't have a complete answer. Okay, so more now we're moving to pitfalls. Feel free to interrupt me anytime. Yeah.
If you have in those networks, you're just saying if you have somehow the right network, I don't have a feel for if it weren't for the network, the way that the data itself is being modeled. I mean, do you think you would get a good fit? Like, is it all being driven by the fact that the network's wrong, or is like the way that the kind of the way the data is, connected to the model itself?
So there yes and yes. So yes, I think that the network does not have enough complexity. And this is linked to one assumption that the method we used, one assumption it has is that it only considered networks where the cycles don't overlap. If we have more than one, here, this reticulation creates a cycle here. And so once I have this reticulation, I cannot add the other one here because it would create a cycle that overlaps, and this method cannot handle this. And perhaps we also need the reticulation that's from here to there, and again, it would overlap, and it's not allowed by this method. So maybe it's just that we don't have enough hybrid edges in the networks. It could also be that just some assumptions are violated, and that's why it doesn't fit. And there could be a number of reasons why assumptions are violated. Yeah. Thanks. All right. So more pitfalls. So what I've been interested in is what is it that we can identify about the network? Phylogenetics originally was on trees, and the answer to this question was really easy. If you have data, you can identify your tree, maybe not the root, period. Nice. Even just from distances between taxa, we can get the tree, we can identify the tree. Not the root, but everything else, yes. For a network, the answer is much more complicated. And for a long time, people forgot to ask the question because the answer was so simple with trees, and they didn't ask the questions about networks. So the answer about what can we identify obviously will depend on what data we're using and then what model we're assuming. So the typical data types that we're using right now are really summaries of our DNA sequences. And the reason is methods that use the full DNA sequences, they're so slow. They can only handle like 3 species. That's it. So we're not really interested in that question yet because we don't have methods that can handle the full data. Methods that people are using summaries, like these quartet frequencies. We take a subset of 4 species and we look at gene trees and what are the frequencies of these gene trees. Okay, we take a subset of A, B, C, D, and then there are 3 options for gene trees. What are the frequencies? Genetic distances could be another like if you think of neighbor joining, this is what it's using. And then the methods that are based that tree admix tools and tree mix are using are really F4 statistics, which are completely equivalent to F2 statistics. And on trees, they're the same as genetic distances, kind of. And on networks, they are very similar. They are variances, basically. And then we can consider different models. Basically, do we assume that there could be incomplete lineage sorting or no incomplete lineage sorting? And so this question was tackled in the work that I started. Oh my gosh, it's almost 10 years ago. Oh. Okay, and then there was a flurry of results more recently about this. So for a long time, we only had very limited results about what is it that we can identify. Let me tell you a little bit. So first, when I talked about average distances, genetic distances, what I mean by this is imagine that we have a network like this, and I'm talking about the distance between taxa C and D. So depending on which place in the genome I'm looking at, I might be in a place in the genome where the history went through this, and then it's a short distance. And then maybe in a different place in the genome, it was inherited through here, and then the distance is much longer. And other places in the genome might have this distance. So if we average across all positions in the genome, we get a weighted average of all these distances. And so that's what I mean by average genetic distance. And I'm assuming we're in the best case scenario, we have really quality DNA data, infinite. We can get this from data perfectly. And the question is, from this perfect data, can I tell which network is a good one? And so first, yes, on a network, we could have something like this, right? Where this is a species merger. Imagine that there was one species, glaciation, separated into two, they speciated, they went back together, da da da. Or there was some ancestor that went extinct. But, you know. So first, from the different data types that I listed, there's no way we can recover what's above this node here. There is a technical term for this node. It's called the least stable ancestor graph theory. Okay. Above this node, there's no information. We have nothing if we use the previous data types. And the position of this node also is not identifiable. So that's known for trees. You know, I said the tree, we can get it perfectly, except for the root. That's what it is about. For networks, that was not proved yet. And so this network is kind of weird. It's not typical. It's a network that there's no roots. But still, we know the direction of these hybrid edges. We still know the direction of that point to admixed species that receive genetic material from more than one parent. We know their direction. The others, we don't know, because we don't know where the root is. So we can only tell that the roots cannot be below a hybrid node. The root cannot be here, the root cannot be here, but the root can be anywhere else. And we cannot tell from our data. That's a semi-directed network that is really lacking graph theory for. So it's taking me into different directions. Something that is not identifiable from average distances and F4s is the age of these reticulation. When did this event occur? When? Well, we cannot tell. We just don't, the information, we can have perfect information, we can have an infinite number of genes, each one infinite size, we can still not tell. So I can imagine this is a zipper. You can zip it up like this, or you can zip it down, all the way down, it would give us the same data. And we can slide, so the zipper is not a good analogy for this, but we can slide the reticulation left and right. This is slid to the left, this one is more slid to the right. Also, that's not identifiable. So there are 3 branch lengths here. This branch T1, T2, and this branch T. We can only estimate one composite parameter. This one for, if we use pairwise distances, if we use F4, there's going to be a squared, gamma squared. Not exactly the same, but one composite parameter. From quartet frequencies, we can get more information, but still some edges are not going to be identifiable. So that's something new compared to trees. And that's something that's hard to convey when we write code that then is going to output some network. And then the biologist will get the results, and it's hard to convey that some of the things are not identifiable. So for inference, what software does typically is to impose constraints. And one option is to say, well, I'm just to constrain these two, it's going to be zipped up. That's it. Also, what that means is, if I zip that one up and this other one up, I get that. So I cannot tell the difference between these two networks. Their zipped up versions are exactly the same. So the data cannot help us distinguish between these two networks. That's one consequence. What is identifiable, so that's a positive result, is what's called a tree of blobs. So now that's a network. I don't know where the root is, but I know what are the reticulations and the admixed populations. So that's a blob, that's a blob, that's a blob. I'm not giving a precise definition, but you can see roughly what this is. The tree of blobs is take each blob and contract it to a single node. So this big thing here gets contracted into this node. This big thing also gets contracted into this node. And you can see the correspondence between edges because I colored some of them. So this tree, it has to be a tree because blobs are, the parts are not tree-like. This is identifiable from the data. This is really cool. Ha. I want to go back to this example I showed before. Sorry, it's going to be really big. This is just one big blob. It's hard to see, but it's all one big blob. So when the tree of blob for this thing is just a star. So the fact that the tree of blob is identifiable, it's not useful at all. Because this network is just too complex, just one big blob. So I mean, this is really cool that the tree of blob is identifiable, but in many cases, it's not really helpful. Okay, but still, okay. So if reticulations are far apart between closely related species, there should be enough structure in this tree of blob for that to be useful. Things are back to the negative results. From average distances, if there is a small substructure in this network that looks like this, we cannot distinguish from a single edge. All this structure is completely invisible. We cannot recover it. Same thing if we have 3 nodes here, then all these reticulations, we cannot identify them. It's undistinguishable from something that has no reticulation whatsoever. And then we can also generalize to more substructure that are connected to the rest of the network through 4 nodes or more. So for example, what that means is here I cannot identify the roots. Okay, so the most I can identify might be this. But here I have one of these substructure that has 3 nodes, 1, 2, and 3. Okay, so it's like this case, 1, 2, and 3, A, B, C, D. And I can contract it to just remove all the reticulations because they're not distinguishable. So 1, 2, and 3. Okay. I would get exactly the same data, not distinguishable. But now I have another triangle, 1, 2, and 3. It's not distinguishable from this. So all in all, this network would give us exactly the same data as that tree. So all these reticulations in this network are not detectable by methods that use distances or F4 statistics. It's good to know, right? Even if it's a negative result. So that's hard. Another example, these 2 networks on top and bottom, they're not distinguishable from one another. All these little complicated things, not distinguishable from that here, at least using quartets and pairwise distances for sure. So they both have the same tree of blobs. They have to because that's identifiable. But distinguishing the details inside blobs is much more complicated. If we assume it's level 1, meaning that every blob is a nice simple cycle, then there are many results on these networks. And most of these results are positive, saying, yes, we can identify things. So these cycles cannot be too small. Okay. Not triangles, or it could be hard to distinguish. And 4 cycles, depending on the data, depending where they're placed, we might not be able to identify everything. But for the most part, we can identify networks if these cycles don't overlap. And that's the reason why one of the methods assumed that the cycles don't overlap. So one of our recent papers I was super excited about is that beyond level 1, if we have something complex here, we can identify the order in which the taxa come around. Maybe we cannot identify everything inside, but at least if the order is well defined, we can identify the order. So that's the first positive, really generally positive result about networks beyond assuming that these cycles don't overlap. It's hard to get more than this, but yeah. So there's really a disconnect between the theory and the practice. I showed you the theory, and maybe I bored you about the theory, about, things are hard to identify. Even if we have perfect data, there are lots of things maybe we can never tell. And yet, in practice, there's so many of these nature and science papers that have these networks. They're definitely not level 1. I mean, here, there is one big blob here, one big blob. This edge is identifiable. It's part of the tree of blobs. This edge also part of the tree of blobs. But this big thing, it's a big blob that has more than one reticulation. It's complex. I'm not sure that there is not another network that would have exactly the same likelihood as this one. That would lead to exactly the same data. And in practice, well, you get software that spits out networks and you just take what it spit out, right? And you publish this in Nature. It's cool. Yeah, there's still a lot we don't understand. So that's the pitfall. There is a really cool paper by the people who develop admix tools and from which many of these networks are published, where they do find this issue empirically, not with theory, but more empirically. And they say there was a reproducibility crisis in association studies, kind of maybe here too. And so we propose a tentative protocol. And basically they say, well, maybe graphs are not stable, like you could get another graph that's quite different, but fits the data just as well, or almost as well. And different complex genetic histories that can result in the same expected, likelihood. But they found this empirically, they didn't have theory. And so they say, well, in that case, use other methods to distinguish between these candidate networks. And basically, we would need to use more data, not just summary statistics. But these methods are super slow. They just don't scale. It's not practical. So that's pitfalls. Other pitfalls is related to the question that Karen asked is, what other thing could make the model not fit? And it would be violation of assumptions. And in my experience, it's really easy to use an extra reticulation to explain something that is really coming from an assumption that's violated. And so one of the an assumption that is made by the F4 statistic is that there's a molecular clock and that all the genes evolve at the same rate in all the populations. And perhaps at the population level, it's a reasonable assumption. But then people are using these methods across species, where maybe this assumption of equal rates across all lineages is not so good. And so one study by Lauren Frankel in the Botany department looked at what is the impact of rate variation. So if you violate this assumption of a clock, it can really lead to, oh, yes, there is reticulation. Well, no, it's not reticulation. It's just variation in the rate of evolution. So that's another pitfall that could be studied some more. Okay. So that's our simulation. And I was saying it's bad, but it's really bad. I mean, there is no reticulation. And if there's rate variation, you could say, yes, there is reticulation almost with a 20% chance. So that's really far from the 5% rate, the type 1 error rate that we would be okay to tolerate. 20% is just too much, or 25%. And with some methods, it jumps to 80%. You're like, I know, I can't tolerate this type 1 error. So we looked at a bunch of reasons for rate variation. It was really rate variation across populations that was an issue. Rate variation across genes was fine. Rate variation across populations, so really a violation of the clock, that was the issue. So trait evolution, how much time do I have for this? Not much. Should I just skip this? Yeah, let me just skip. Well, no, we have 7 minutes. So trait evolution is about answering these questions. Now we have a network, we have a tree, and we ask questions about specific traits we're interested in. It might be flower color. It might be the pollination system. It might be head length or toe pad width, And we might be interested in the correlation between these traits, or how they change in some groups. Flower color evolves very fast compared to other groups where flower color does not evolve very fast. We might be interested in adaptation. Was there a shift overall in some clades to adapt to a different optimum? So most of the models for trait evolution will use Brownian motion if it's neutral for Stein-Uhlenbeck process, if we want to model natural selection, or Markov processes for discrete traits. And so we want to, everything that we know how to do on a tree, we want to be able to do this on a network. And again, the paradigm shift says that we can ask new things on a network. We want to ask all the previous questions and ask new questions. For example, is there a transgressive trait evolution where in an admixed population, you get something that's not the average. You get something that maybe is outside the parents. So yeah, so we can answer these questions. That's another cool example where here, again, these baobab species, and here you have flower color, and there is some support, not strong, but some support that flower color was transmitted through reticulation. So that's something interesting to know. It's not convincing, but it's and then accounting for the network also changes our ancestral reconstruction of ancestral states. So if we account for the reticulation, we have a much more higher posterior probability that the ancestor in Madagascar had yellow flower. So if we only use a tree, that's a 50% posterior probability. If we use a network, it's a 70% posterior probability. So that has quite a big impact on ancestral state reconstruction also. Let me stop here so that there's some time for questions. Oh, no, I want to, oh, this is very cool. Thank students who worked in my lab and collaborators. It's really fun to work with everybody. Thanks. Yeah.
So, question, do you recall when they first identified or tentatively introgression between Neanderthal and human genomes, how they excluded the hypothesis of incomplete lineage sorting? Or did they use an asymmetry argument like the one you presented, or was it based on something else?
No, so I think that's a great question. So the question was when they first identified introgression from Neanderthal into modern humans. So they use methods that use edge lengths. So you go through the DNA sequence and you look at how within a window, what is the average distance or difference between let's say modern humans and Neanderthals, right?
And they split the modern humans into three or four populations in comparison. So
Yeah.
But I don't recall. When you say edge lengths, you mean edge lengths with just genetic distance or with respect to some other metrics?
So it would be measured using genetic distance, right? And can I go back? Yeah, using the genetic distance, but here within using a sliding window. So you don't average across the entire genome. You see that in some places in the genome, there's a really short distance. So some genes are at a really short distance between humans and Neanderthals. Whereas for most 98% of the genome, this distance is much bigger.
Yes, wouldn't lineage sorting give you the same path?
Ah, this is a great question. No, it would not. For genetic distances, it doesn't. So what happens if there is? Yes, yep. So here with incomplete lineage sorting, A and B are quite distant. If they don't coalesce, they have to be very distant. And C and D, if that's because of ILS, they are very distant. Whereas if it's from reticulation, C and D here would be fairly closely short. The branch lengths would be fairly short in the gene tree.
So then what's the advantage of using these asymmetry arguments as opposed to distance measures to distinguish between the incomplete lineage sorting and the gene sorting?
That's a great question, because so the question is, what's the advantage of using this asymmetry argument versus distances? So distances, that is affected by mutation rate. So if there is, we can explain a short distance here by a change in the mutation rate where maybe just the mutation rate was very slow. They coalesced a long time ago. C and D coalesced a long time ago, but then the mutation rate could have been really slow on these two branches. And then if we allow for variation in the mutation rates, then we don't know how to use the distance information. And the asymmetry is more robust. The asymmetry argument does not make an assumption about mutation rate variation.
Closely related species like humans, the anthropological distance are
Yes,
phylogenetically distant species.
exactly. At deeper time scale, it's the asymmetry argument is better. And at shallower time scale, using distances is probably fine. Yeah. And more informative. So same thing, if we wanted to detect a merger, where we might take two individuals from that population even. Let's say there was glaciation, these two populations split, and then glaciers melted and they come back together. We would be able to detect this using distances, because for much of the genome, the distance between these two individuals would be short, or even like this. And then for some proportion of the gene, so mostly in here, right? And for much of the genome, then if the distance, no, for a small proportion of the genome, the distances would have to be much wider. So the distance would have a bimodal distribution, where for many genes, it would be a shallow distance. And for some genes, it would be a much longer distance. We would see on the gene tree. But if we don't use distances, we have zero information about that event. But that could also be explained by some genes evolving very fast compared to the others. How do we tell? I don't know.
We're actually at the end of our time, so please, big round of applause for Professor Ané.